today's session, photosynthesis part 3. Great. So welcome you all the learners, myself Swati Pawar, one of the Biology Plus educators on Unacademy platform. Welcome you all the learners for today's session, that is photosynthesis part 3. Okay, so we are going to start for the session. There was some network issue, so it got delayed 3 to 4 minutes. So we will start for the session right now. Before going to the main session, as usual, you know that all the concepts or everything is given in the description box. All the links and important what you can say information. So again, before that, this is a plus course, right? So Swati Live is my code to use the plus subscription code. So premium content at affordable price, right? So what are the features of your plus course? Plus course features on Unacademy platform is nothing but you have a premium content on affordable price. We have structured live courses, dedicated live doubt solving sessions. We do have quizzes, then batches are there for complete preparation, right? Just a minute, just check it. Just a minute. So we do have these features. Then we are also having different batches like Hindi, Marathi, English. We do have different batches for your plus course. So we have 1 month plan 2,700. We have 6 months plan and 12 months plan accordingly. That is 12,150 and 14,850. New batches are already started. You can start enrolling for yourself. Till MHT CET you will be prepared with lot many leaderboards, mega quizzes, weekly quizzes and analysis session as well. Clear? So great. These are the educators who teaches you in the batch courses. You can see my name here, Biology Swati Pawar, right? Biology. Great. So shall we start for today's session? So our today's session is dark reaction. In previous session आपण शेवटच्या 2 सेशन्स मध्ये लाईट रिॲक्शन कंप्लीट केलेली आहे. आपण लाईट रिॲक्शन मध्ये आपण बघितलेलं आहे की हाऊ द नॉन सायक्लिक सायक्लिक फोटोफॉस्फोरिलेशन टेक प्लेस, हाऊ द एटीपी इज जनरेटेड with the help of electron carriers. Then what is photolysis of water, why there is a need of photolysis of water. So these all important things we have already studied in the previous sessions, right? So today we are going to start with the dark reaction. So generally dark reaction occurs in the stroma. As I said, there is a difference that there are 2 reactions. One is light reaction and another one is dark reaction. Another one is dark reaction, right? Dark reaction. So in light reaction take place in thylakoid, that is in grana of chloroplast, whereas dark reaction take place in stroma, which is a granular substance in the chloroplast, right? Because stroma contains all the enzymes which can break the complex compounds into the simple to give rise to glucose. So why we do dark reaction? What is the motto of carrying out dark reaction in the stroma? To prepare glucose, to prepare glucose. And what is the motto of carrying out light reaction in plant? To generate energy, to generate energy, ATP energy, right? ATP energy. So these are the 2 things different for light and dark reaction. So now how the glucose is prepared in the plant body. So Kelvin cycle is very important. Now you must have seen that there are so many cycles in dark reaction. You have C3 cycle, you have photorespiration, you have C4 cycle, you have CAM plant. You take any plant, तुम्ही कुठलंही plant घ्या, ते जे तुम्ही बघताय हे ज्या सायकल्स आहेत C3, C4, कॅम किंवा फोटोरेस्पिरेशन, या सायकल म्हणजे सगळे प्लांट एकाच ह्याच्यातले आहेत. बट होतंय काय, availability of CO2 or O2. Right? So depending upon the habitat of the plant, whether the plant is growing in the dry and arid region, whether the plant is growing in the environment where oxygen is more, CO2 is less, or whether the plant is growing where CO2 is more, O2 is less. So depending upon this concentration, what is happening? Yes, the cycles are taking place. Clear? So if more good quality of CO2, if good quality, if there is a lot of CO2 in the atmosphere, or moderate, like whatever the CO2 which is required for the plant for photosynthesis, if that appropriate amount of CO2 is present in atmosphere. Yes, good morning GD. So a lot of CET is there, so CO2 is there. So in atmosphere, if CO2 is present in an appropriate quantity, then C3 cycle take place in plant. So C3 cycle take place in plants. Clear? If CO2 is less and O2 is more in the atmosphere, then that only plants will modify themselves into some different, as photorespiration, where CO2 is lost. We are going to see the cycles. Third one, what is third condition? Third condition, if any plant is grows in dry arid region, grows in dry climatic region, right, or desert region, then what happen? Then water is less, so CO2 or the stomata remains closed at the daytime to check the transpiration process. So desert region or arid region, water is less or decreases. So what happened because of this? Stomata remain closed. Stomata remain closed. Stomata closes during the daytime, right? And it opens during the night time when the environment soothes, so that they can fix the CO2 and start their cycle. So this is one more cycle which is called CAM plant, crassulaceous acid metabolism. Clear? So this is another type of plant which undergo modification. There is a fourth condition. There is fourth condition that some plants have two type of chloroplast. Some plants have two type of chloroplast, right? So one is mesophyll and another is agranule chloroplast. If any plants are having two type of chloroplast, then that type of plant modified themselves or get adapted themselves into a new cycle called C4 cycle, or it is also called HSK pathway, Hatch-and-Slack pathway, where CO2 is fixed twice. One time the CO2 is fixed in the mesophyll, where bundle of chloroplast is there, and the second type where the chloroplasts are not there, but there a Kelvin cycle can take place in the stroma. So CO2 is fixed twice in the C4 cycle, which we are going to see right now. Okay? So this was a basic concept about the dark reaction. The dark reaction take place in stroma because we need to prepare the glucose. So फंडा काय आहे की कुठलीही सायकल असो, या चार कंडिशन मधली कुठलीही कंडिशन असो, plants are modifying themselves, adapting themselves to survive, but सगळ्यांचा मोटो एकच आहे की everybody is going to end up in Kelvin cycle. Why? Because Kelvin cycle is the one in which glucose formation is going to take place. Am I right? So प्लांटला कुठल्याही प्रकारचा मॉडिफिकेशन करू द्या, त्याला सर्व्हाइव्ह करण्यासाठी त्याच्या कंडिशन प्रमाणे तो मॉडिफाय होणारच आहे, बट त्याला फायनली कुठे जावं लागणार आहे? C3 सायकल मध्ये जावंच लागणार आहे. तुम्ही कुठलीही सायकल घ्या, दे फायनली हॅव टू एंड अप इन द C3 cycle, that is Kelvin, because Kelvin is the one in which glucose is prepared in a systematic manner. So you take photorespiration plant, you take C3 plant, you take CAM plant, or you take C4 plant, you take any plant. Primary first step will be different, but next step will be compulsorily same in everybody, that is C3 cycle. They are going to enter into the C3 cycle to prepare glucose. So they all are going to show common cycle, Kelvin cycle. That is, the Kelvin cycle is the one which is useful for preparation of one glucose molecule, C6H12O6. So let's start. Now first reaction. This is also called independent reaction. Dark reaction meaning what? Now this take place in stroma, right? These all cycles, whatever I have given, these all cycles take place where? In the stroma. So these all cycles do not require light now, because we have already completed light reaction and then we are moving into the dark reaction. Because first in the light reaction what we are doing? We are generating ATP energy. नाव वी डू हॅव एनर्जीज. प्लांटमध्ये एनर्जी आलेल्या आहेत, एटीपी आहेत, NADPH2 आहे, सगळं आहे. मग आता तुम्हाला काय करायचंय म्हणजे प्लांटला या एनर्जीला आता युटिलाइज करून ग्लुकोज प्रिपेअर करायचा आहे. आणि तो ग्लुकोज कुठे प्रिपेअर होणार आहे? येस, स्ट्रोमामध्ये. आणि लाईट रिॲक्शन कुठे झाली? थायलॉइडमध्ये. बरोबर. मग आता थायलॉइडमध्ये ज्या काही एनर्जीज फॉर्म झालेत त्या सगळ्या आता कुठे येणार? स्ट्रोमामध्ये येणार. बरोबर. आणि स्ट्रोमामध्ये कुठली रिॲक्शन होणार आता? डार्क. वाय इट इज कॉल्ड डार्क? बिकॉज इट इज लाईट इंडिपेंडेंट रिॲक्शन. इट इज व्हॉट? लाईट इंडिपेंडेंट रिॲक्शन. मीनिंग हिअर नाव देर इज नो रिक्वायरमेंट ऑफ लाईट ॲट ऑल. कारण की आपल्याला लाईटची रिक्वायरमेंट का असते? टू प्रिपेअर द एनर्जीज. बट आता ऑलरेडी एनर्जीज आपली पहिली स्टेपमध्ये लाईट रिॲक्शन फॉर्म झालेले आहेत थायलॉइडमध्ये. फक्त आपल्याला त्या एनर्जी थायलॉइड मधून घ्यायचे आणि त्या आपल्याला स्ट्रोमामध्ये युटिलाइज करायचे बाय प्लांट. दॅट इज नॉट अस. Plant is going to utilize that light reactions in the stroma and they will start preparing the glucose. But now glucose is prepared by fixing CO2 and water molecule. And what will be your side product? Plant's side product काय असेल? C6H12O6 plus 6O2 will give you 6CO2 will give you C6 that is 6CO2 plus 6H2O will give you C6H12O6 plus 6O2 or energy, right? So this is a balanced chemical equation of the photosynthesis where CO2 and water is fixed in the stroma and glucose is formed and O2 is a byproduct. Clear? Am I clear this concept? So if I want to prepare a glucose, plant is going to fix 6CO2 because one glucose molecule takes 6 turns of the, right? Then I will add up water molecule. I will add up water molecule with this and I will get 6H12O6 plus 6O2 as a byproduct. So this is a byproduct of the plant and this is a glucose which we got. Clear? And here this all thing is going to happen in presence of ATP energy which was formed in light reaction. Am I clear? So light reaction मध्ये ज्या ATP फॉर्म झालेल्या आहेत त्याच्या प्रेझेन्स मध्ये CO2 आणि वॉटर मॉलिक्यूल फिक्स होणार स्ट्रोमा मध्ये and glucose will be prepared. So we'll go for the first cycle, that is Kelvin cycle. Right? So Kelvin cycle is also called C3 pathway. It is also called which cycle? C3 pathway. Why it is said to be C3 pathway? Now बघा तुम्हाला सायकल्स थोड्याफार बायहार्ट कराव्या लागतात. बायहार्ट नाही बट तुम्ही 2 वेळा जरी बुक्स मध्ये प्रॅक्टिस केल्या त्याचा कॉन्सेप्ट समजून घेऊन तर तुम्हाला फार इझी आहे. क्लिअर? सो यू हॅव टू just go through twice or thrice. You have to practice it in your book and you are done with that. Clear? Yes. Garki, welcome. Welcome. Good morning. Good morning, beta. Chalo. So great. C3 pathway. It is given by first time by Melvin Kelvin. Okay. Melvin Kelvin is nothing but it is the scientist name. He has done first time. त्याने Kelvin cycle. Melvin Kelvin ने पहिल्यांदा ही सायकल केलेली कशामध्ये? By using chlorella, that is a blue-green algae. So he has considered an chlorella algae, and in that he has done for the first time experiment by reacting that with a radioisotope material. Right? From that he came to know that yes, in chlorella when he fixed a heavy radioisotope of CO2, the Kelvin cycle has completed, leading to the preparation of the glucose molecule. Right? Yes, great. Correct. So C3 pathway is given by Melvin Kelvin. Am I clear? 1951. Right? So what he has done, he has converted the carbon dioxide and water into the glucose by applying these features on the chlorella for the first time. Great? Yeah. Chalo. Next one, product of light reaction. Now product of light reaction. What was the product of light reaction? ATP and NADPH2. Now these energies are going to get utilized for fixing CO2 and water molecule to prepare glucose. So as I said, the reaction, you have to fix CO2 and water molecule to get glucose and oxygen as the byproduct. Am I clear? Chalo next. Kelvin cycle is divided into 3 steps. Kelvin cycle ही किती स्टेप मध्ये झाली डिवाइडेड? 3 स्टेप. You can see here the first step is carboxylation, second is reduction and third is regeneration step. Am I clear? Yes. So first step is carboxylation, second is reduction and third is regeneration. Now what do you mean by the first step carboxylation? कार्बोक्सिलेशन मीनिंग नाव फिक्सिंग CO2. आपण डार्क रिएक्शन मध्ये सगळ्यात पहिले प्लांट काय करणार आहे? आता CO2 ला फिक्स करणार आहे विथ द वॉटर मॉलिक्यूल. सो इन द स्ट्रोमा who is there to accept that CO2 molecule from the atmosphere? Yes, exactly. CO2 is going to get fixed up by RUBP. That is, yes. So it is ribulose biphosphate. So ribulose biphosphate is nothing but it is an enzyme, Rubisco, which combines with RUBP component, that is reactant, to give rise to what? Yes, PGA. So PGA is a first stable compound. Okay, first stable compound. Clear? So CO2 is fixed now. Great. So 6CO2. Now one glucose molecule चा formula काय? एक glucose molecule चा formula काय? C6H12O6. बरोबर. सर आपल्याला CO2 किती वेळा फिक्स करावा लागेल? 6 वेळा. कारण की त्याचा फॉर्म्युला काय आहे? C6H12. सो आपल्याला 6 वेळा CO2 फिक्स करायचा आहे. तर डायरेक्टली तुम्हाला ही एक ग्लुकोज मॉलिक्यूलची सायकल दिलेली आहे. एक ग्लुकोज मॉलिक्यूलची सायकल दिलेली आहे. सो CO2 is for the first time fixed by what? Yes, it is fixed by RUBP, right? So RUBP is fixed by CO2 to give rise to unstable compound called keto acid. Now this unstable keto acid is going to get converted into the simplest form, that is called PGA, phosphoglyceric acid, which is the simplest form. So you are going to get 6 PGA, that is 2 molecule of PGA, 3C 3C each. 3C 3C each. बरोबर ना. इथे 6 कार्बन ॲटम आहे. मग या 6ला तुम्ही स्टेबल मध्ये 2 3C 3C मध्ये कन्वर्ट करणार. बरोबर की नाही? मग त्या 2 3C 3C मध्ये तुम्ही कन्वर्ट केला तर तो 3C वाला कंपाऊंड कोण असतो? PGA असतो. आता हा जो 12 आकडा दिलाय तुम्हाला तो पूर्ण तुमच्या एक ग्लुकोज मॉलिक्यूलचा 12 आकडा दिलाय. बरोबर. पण आपल्याला एक CO2 जेव्हा फिक्स होतो तेव्हाचा PGA मॉलिक्यूल किती आहे? 2 मॉलिक्यूल्स ऑफ 3C 3C इच. म्हणजे एक कार्बन डायऑक्साइड मध्ये तुम्हाला 2 PGA फिक्स होतोय. म्हणजे C6H12O6 6 वेळा CO2 फिक्स केला तर किती PGA मिळतील? सिम्पली 3 × 6 विल गिव्ह यू 12. मग तुम्हाला हे कंप्लीट एक ग्लुकोज मॉलिक्यूलचा फॉर्म्युला दिलेला आहे. क्लियर? ग्रेट. सो दॅट इज कॉल्ड कार्बोक्सिलेशन वेअर CO2 इज फिक्स्ड in the stroma and CO2 is accepted for the first time by RUBP. RUBP is going to accept that CO2. It will give you first unstable keto acid. Then that unstable is broken down into the 2 stable compound called as PGA, 3C 3C each. So in one carbon dioxide you are going to get 2 molecule of PGA. So you have to multiply, that is 2 × 6. So you get what? 12 PGA molecule. Clear? Great. Now second one. Second, reduction. Now your second process is what? Reduction. Reduction meaning काय आहे? This PGA will get reduced. This PGA will get reduced to some other compound. So PGA is going to get converted into a next compound called PGAL. That is phosphoglyceraldehyde. You can see here. Correct? So from this 12 molecule, only 2 molecules of PGAL will go in the synthesis of glucose. Whereas remaining 10 molecules will go back for regeneration of RUBP. बरोबर ना. कारण की पुढचा CO2 फिक्स व्हायला तुम्हाला RUBP लागणार आहे. मग RUBP फॉर्म करण्यासाठी तुम्हाला काही मॉलिक्यूल रिजनरेशन मध्ये टाकावेच लागतील. बरोबर की नाही? Yes. So this third point is regeneration. Clear? So first is carboxylation, second reduction, and third regeneration. Am I clear? So reduction is nothing but PGA will get reduced to PGAL, that is phosphoglyceraldehyde, and then next 10 molecules will go for regeneration. Why it is called 3B? Because 3A process is synthesis. 3A process is synthesis of glucose molecule. So basically you have carboxylation, reduction, then synthesis and regeneration. So you have 4 steps basically. Clear? Third step is having 2 forms. That is one synthesis and another regeneration. Clear? Very good. Great. So chalo, 2 molecule of PGA will go for formation of glucose. So first compound from PGA you will get fructose 1,6 diphosphate. Now we have to reach till glucose. So we have to remove the phosphate group slowly. So from fructose 1,6 diphosphate, I am going to remove one phosphate group, which is called dephosphorylation. Dephosphorylation. Clear? So fructose 1,6-diphosphate will be, by removing one phosphate group, will reduce to fructose 6-phosphate. fructose 6 मधून परत मी एक phosphate group काढणार का? नाही, अजून त्याला टाइम आहे. fructose 6 will get it converted into the isomer called glucose 6-phosphate, and now I'll remove one more phosphate here, dephosphorylation, to get glucose molecule. So fructose 1,6-diphosphate, fructose 6-phosphate, glucose 6-phosphate, and then glucose finally. Clear? So synthesis, glucose is formed. Now 10 molecules are going back for the regeneration process. Clear? So regeneration मध्ये काय होणार? Now PGL, 10 molecules मध्ये PGL will get converted into some other compound by adding one CO2 here also, one carbon atom here, to get converted into erythrose. Then in erythrose, more 3 carbon atoms are added to get converted into pseudoheptulose. Pseudoheptulose will remove 2 carbon atoms to get converted into 5C called RUMP. Then in RUMP again you are going to add one phosphate group to get converted into RUBP. So here one ATP is utilized to convert RUMP into RUBP. Clear? So ribulose monophosphate now get converted into biphosphate, and now biphosphate is ready to fix next CO2 for the next cycle. Am I clear for this? Yes. So this was Kelvin cycle. So now my question is, for 1 Kelvin cycle, for one CO2 in Kelvin cycle, for fixing 1 CO2, how many ATP energies are required? So we need 3 ATP and 12 NADPH2. 12 NADPH2. This is for 1 CO2. Now my glucose formula is what? My glucose formula is C6H12O6. Am I clear? So I am fixing CO2 6 times. So now if I take 6 CO2, for 6 CO2 I need 18 ATP and 12 NADPH2. Sorry, this is 12 NADPH2. Right? So for 1 CO2 I need 3 ATP and 2 NADPH2. So for 6 CO2 I need 18 ATP and 12 NADPH2. You will say now how 18 ATP? I'll tell you, and how here 3 ATP. So look here. See, in this reduction part we have 12 ATP. Plus for each carbon atom we have to reduce 1 ATP. We have to lose one phosphate group from ATP to ADP to convert RUMP into RUBP. So 1 ATP in each carbon dioxide here and 2 ATP in each carbon dioxide here. So total 3 ATP for 1 CO2. So for 6 CO2 we need, yes, 18 ATP, 12 here and 6 this side. So 12 and 6 is 18 and 12 NADPH2. Clear? So this was an Kelvin cycle from where the glucose is prepared. Clear? Chalo, we will go for the balance sheet I have given you. So 6 CO2, 1 glucose molecule we are going to take. How much ATP? 18 ATP and 12 NADPH2, right? So this is your, 1 minute, this is your total ATP for Kelvin cycle. Total ATP for Kelvin cycle. Okay? That is for 6 CO2. For 1 CO2, you have to fix 3 ATP and 2 NADPH2. 2 NADPH2. Okay? Chalo, next one. Now, one more we will do, and then we can stop for the session, because as it is we are going to end up the session, our time is up, but still photo respiration one more which is linked to that. Now hope Kelvin cycle is clear. It is very easy. Twice you are going to practice and you are gone through it. So ATP चे कॅल्क्युलेशन जे असतं ते लक्षात ठेवायचे तुम्हाला केल्विन सायकल मध्ये. म्हणजेच एक CO2 ला 3 ATP आणि 2 NADPH2 ची गरज आहे. म्हणजे 6 CO2 ला फिक्स करायला आपल्याला 18 ATP 12 NADPH2 ची केल्विन सायकल मध्ये गरज आहे. सो बॅलन्स शीट इम्पॉर्टंट आहे सगळ्या सायकल साठी. ठीक आहे. नाउ व्हॉट इज फोटो रेस्पिरेशन? आता केल्विन सायकल मध्ये ग्लुकोज फॉर्म झालेला आहे. करेक्ट? सो फोटो रेस्पिरेशन मध्ये काय होतं? फोटो रेस्पिरेशन हेच प्लांट असतात, केल्विनचेच प्लांट असतात, राईट? C3 प्लांट्सच असतात. बट नाउ C3 मध्ये काय होतं? CO2 वॉज इन अन अबंडंट इन द ऍटमॉस्फिअर. CO2 एका गुड अमाउंट मध्ये प्रेझेंट होता ऍटमॉस्फिअर मध्ये. सो RUBP ने त्याला फिक्स केला आणि सक्सेसफुली ग्लुकोज मॉलिक्यूल हा फॉर्म झाला. बरोबर? बट फोटो रेस्पिरेशन मध्ये काय होतं? आता फोटो रेस्पिरेशन मध्ये समजा काही C3 चे प्लांट अशा एरियामध्ये आहेत जिथे CO2 चं प्रमाण कमी आहे ऍटमॉस्फिअर मध्ये आणि O2 चं प्रमाण जास्त आहे. सो RUBP इन्स्टेड instead of fixing CO2, it will fix O2. राईट, दॅट इज कॉल्ड ऑक्सिजनेशन. सो इथे कार्बोऑक्सिलेशन होतं, इथे ऑक्सिजनेशन असतं. मग अर्थात प्रॉडक्ट सुद्धा वेगळे वेगळे असतील फोटो रेस्पिरेशनचे. पीजीए जो तुम्हाला केल्विन मध्ये मिळाला, पीजीए तो तुम्हाला फोटो रेस्पिरेशन मध्ये नाही मिळणार पहिला स्टेबल कंपाऊंड. राईट, इट विल बी समथिंग एल्स बिकॉज नाव वी आर नॉट फिक्सिंग CO2, वी आर फिक्सिंग O2 with the RUBP. Clear? So this is your photo respiration cycle. So how this plant adapt themselves? फोटो रेस्पिरेशन का म्हटलंय, कारण याच्यामध्ये आता CO2 फिक्स नाही होत आहे, O2 फिक्स होतोय. त्यामुळे या प्लांट मधून CO2 बाहेर जाण्याचे जास्त चान्सेस आहेत. आणि प्लांटच्या बॉडीमधून CO2 बाहेर पडणं हे चांगलं नाहीये. मग CO2 बाहेर पडतोय त्याला आपण म्हणतो फोटो रेस्पिरेशन. राईट, फोटो मिनिंग इन प्रेझन्स ऑफ लाईट, रेस्पिरेशन लुझिंग देअर CO2 रेस्पिरेटरी कंपाऊंड, लुझिंग द रेस्पिरेटरी कंपाऊंड. सो हे जास्त प्रमाणात होऊ नये म्हणून या प्लांटने स्वतःला कशामध्ये अडॅप्ट केले, 3 ऑर्गेनल मध्ये अडॅप्ट केले. क्लोरोप्लास्ट, peroxisome, mitochondria. These are the 3 organelles they have adapted themselves. Am I clear? So cycle बघा, आता तुम्ही बघितलं तर RUBP is accepting O2, right? So their first stable compound is glycolate and not glycerate. It is phosphoglycolate. Now why it is 2C? Now why it is 2C compound? 2C instead of 3C it is 2C because we are fixing O2 here and not CO2. So RUBP anyways it is a 5 carbon atom. So 5 carbon atom is unstable, so it will break into 2 molecules, 2C and 3C. So 3C will be PGA and 2C will be phosphoglycolate. So 5C is divided into 2 stable compound. One is 2 carbon atom phosphoglycolate and another is 3C carbon atom phosphoglycerate, same as Kelvin cycle. Clear? So you have got 2 different compound, 2C and 3C for 5 RUBP. So now this cycle we go on in these 3 organelles to preserve CO2 maximum. But still in this cycle 25% of the CO2 is lost from the plant body, lost. So we call this cycle as a photorespiration. Photorespiration. Am I clear? Right? So this plants have modified themselves into 3 organelles to preserve 75% of CO2. So because of this modification, only 25% of CO2 is lost from the parent's body, plant body. Clear? Great. Okay. Chalo, so next one is what? Yeah. So next one, chloroplast, peroxisome and mitochondria. These are the 3 organelles in which this cycle has taken place. So this photorespiration is also called as glycolate cycle. Which is the name of this cycle? Glycolate cycle. Am I clear? So glycolate का, कारण की तुम्ही बघू शकता इथे सगळे glycolate चे प्रोडक्ट आहेत. Phosphoglycolate, only glycolate, then glycoxylate, glycine, serine. So it starts from the glycolate. So photorespiration is also called as glycolate cycle. Clear? So या पद्धतीने photorespiration मध्ये only 25% of CO2 is lost from the plant body. Remaining 75% is conserved back. Right? So but this cycle is again going to enter into the Kelvin cycle as I said. Plant ने कुठलेही adaptation करू द्या, पण finally ते plant कुठे जाणार आहेत? It is going to carry out the Kelvin cycle to prepare the glucose. Am I right? So finally glucose साठी तुम्हाला किती ग्लुकोज प्रिपेअर करायला तुम्हाला केल्विन मध्ये किती पीजीए मॉलिक्यूल होते? 2. आपल्याला फोटो रेस्पिरेशन मध्ये किती पीजीए मिळालेत? एकच मिळालंय. मग अजून एक पीजीए मिळवण्यासाठी ही सगळी सायकल सुरू झाली. सो बघा आता इथे जेव्हा ही सायकल पूर्ण एंड होते तेव्हा तुम्हाला अजून एक दुसरा पीजीए मिळालेला आहे. सो नाव यू हॅव 2 पीजीए, 1 year and 1 year. So now 2 PGAs in the chloroplast can start Kelvin cycle. So now they are going to enter into the same Kelvin cycle, 2 PGA, PGAL, synthesis of glucose, regeneration, everything same. Only difference is instead of CO2, O2 is fixed. Right? So 3 organelles are participating in photosynthesis. So these 3 organelles are going to conserve 75% of CO2. Only 25% of CO2 is lost. And second reason of this modification of 3 organelle is to form the second PGA molecule. Because here only we have got 1 PGA. But for Kelvin cycle, to start Kelvin cycle, we need 2 PGA. So to get that second PGA, we have completed this whole cycle. So now this 2 PGA in the chloroplast will enter into the Kelvin cycle for the production of glucose molecule. तर समजले, कुठलाही मॉडिफिकेशन असू दे कुठल्याही प्लॅनमध्ये, त्यांना फायनली स्वतःला अडॅप्ट करून असं फॉर्म करायचे की ते फायनली केल्विन सायकलमध्ये जातील टू प्रिपेअर द ग्लुकोज. दिस इज द मोटो ऑफ each and every plant. Am I clear? So today we have completed 2 cycle, Kelvin cycle and photo respiration concept. Now tomorrow, after 2 days, I think we are going to cover up MCQs on photosynthesis. So far whatever I have completed the chapter, I am going to put the MCQs on it. So we will see after 2 or 3 days we will be back for the MCQ sessions. So hope you don't have any doubt. Clear? Great. Chalo, so shall we end up the session? We don't have any doubt. Great. So see you in the evening on Unacademy, that is free class, special class that is on Unacademy at 9 p.m. So we will be doing the respiration and circulation part. Am I clear? So next session we are going to meet for the MCQs of photosynthesis, and if you don't want to wait till that sessions, my already free courses and paid classes are going on Unacademy platform. There you can join us and you can start attending the lectures, and if you have any doubt you can ask me. Clear? Great. So shall we end up the session? Good day everybody. Bye bye. Take care. Yes.